Hi, everybody. Great to see you. Thanks for coming. The promise of omic mapping is such an interesting title because we talk about mapping or modeling, and it's like, what is omic? Are there many omics? Is there one omic? So today we have a panel of experts all working in this field, and our goal with this session is to talk about the real potential of this multimodal mapping, the limits, and the value. So I am going to let our panelists introduce themselves and say a few words about what they're working on in this field. So I'll start with Shai.
Hi, everyone. Shai Shen-Orr. I'm the Co-CSO of the Human Immunome Project. My background is in AI and systems immunology. So I'm a computational systems immunologist, if you like, really studying human immune variation and immune aging and its relation to life. And the immune system is all about, basically maintaining homeostasis of the body, maintaining balance that connects everything together. It's a complex system. It changes all the time. And so you need to do multi-omic mapping. I'm based at the Technion, the Israel Institute of Technology, and I co-direct our AI and Healthcare Solutions Center, our Healthy Aging Institute, and I've also founded a pharma AI company that does computational disease modeling. Happy to be here.
Great, thank you. Christina.
Hi, everybody. I'm Christina Lopes. So great to be here and great to be with Jane in this amazing panel. And just to say that this is a really special house because I've been coming to Davos since 2011 and I've not seen a presence of science like this. So I am so happy, number one. Number two, so I'm Christina. As I said, I'm co-founder and CEO of FidoCure. We are taking a cross-species angle. So we look from the genomics lens, but we interrelate clinical data and outcomes to really fight cancer and reimagine animal testing from something that injures animals into actually bringing animals as a patient and learning, because remember, from the data set angle, we built for AI, data is the fuel. And we are able to really have just a huge data set. We don't have HIPAA, which are privacy limitations in the United States. So we can really dive in and generate really interesting hypotheses and then validate them very rapidly. So that's what I'm doing here with this illustrious.
I've got my own. Thanks a lot. Yeah, Thomas Hartung. I have a handful of professorships, mainly in Johns Hopkins in US. My interest is in replacing animal testing by new modern technologies. And out of this, I have developed a particular interest in what constitutes evidence. How can we prove something? And which role do big data, factfulness play, which started me into the AI field. And I'm, for this reason, also the field chief editor of Frontiers in Artificial Intelligence, where we have published about 1,800 articles now.
Great. Okay. So basically, you've got a bunch of pioneers here, both in the omics space and in the AI space. And so I think we could just have a conversation about the title of this session. And, we've talked about mapping. We've talked about modeling. Shai, why don't you kick off with how you define it and how you work with it?
Oh, that's a big question. I will say, first of all, I've noticed that every time I see the title, it keeps changing. So I thought it was multi-omics mapping, but I can go with any omic mapping. I will say that to me, really, if I look at life sciences, this is a field of unknown unknowns. There's too many things that we don't know and we will never know. And actually, every time we find out something really fundamental, we give a Nobel Prize on it and we need to model something new, right? And so the question is, when we look at biology in all of its complexity from different angles, are we seeing the same information? Or are we seeing complementary information? And it's quite clear at this point that not only when we look at different data types, the information is complementary in the half-life that we're looking at, the dynamics of the information, what actually signal we catch, but it's even true when we look at the same data type generated in two different technologies. There's very few technologies that are fully replacing an alternative one. They're actually all complementary, and when it comes to thinking about multi-omic mapping and so forth, I think about really bringing everything to the table to ultimately give me some kind of a triangulation, just like we do with our phones for GPS and so forth. I need to be able to point the state of the system. And so to me, that's the fascination of mapping with multi-omics. And it's a life adventure that will, because of its unknown unknowns, will just continue forever. So I'm having fun.
Okay.
So to stem from that, I mean, you guys are mapping the human immunological system. We're more in the, let's say, less discovery and more drug development side. So we're generating evidence, leveraging the similarities between human genomics, canine genomics, interrelating that with phenotype, with clinical data, with what treatments were given and outcomes. And we're doing that in order to move drugs through the regulatory body in the United States, the FDA. So we're kind of at that end, of the spectrum. So we've been packaging a lot of evidence data sets and actually working with really bipartisan movements at the regulatory body that are welcoming very much data and AI, very much so. So they just actually December 15 lowered the bar for real world evidence and interrelating all of these omics. And so we're very interested in generating that predictive engine of what therapeutic is going to actually have an outsized benefit. But we're based in genomics. So that's how it loops back to genomics.
Right.
I mean, when I refer to evidence-based methodologies, I'm referring more to evidence-based medicine, to systematic reviews, to quality scoring, meta-analysis. That's a different tool set. We have always been using evidence. We're producing evidence, is what every scientist would claim. But there's a different value to being self-critical. And from my point of view, omics was the buzzword which developed out of genomics. Yeah? We want to have all genes. And at some point we said, you only are an accomplished scientist if you have created one ohm of yours. Yeah? And somehow everybody claimed a new ohm. Yeah? Today we mostly call it big data. It's just the same, but big data are interesting multimodal. And this is essentially multi-omics. Yeah? So that's the modern spin on exactly this discussion. Yeah?
Yeah, and I think just to add on that, what's really important is in this era of AI, data is the fuel. And if you think of the aha moment of ChatGPT, right, bringing in text and images from the web and AlphaFold with a lot of the proteins. So what's like the next aha moment, right? So that's kind of one of my interests, that convergence.
Yeah. Yeah, this is a good place. And I think we should do immunome first, and then we'll do exposome.
All right. So I want to go back, first of all, to two things. So one is the promise, right? So I talked about unknown unknowns, but the promise is you see this every day, because as you measure additional kind of high-dimensional data and additional data types, the evidence builds up, to Thomas's point, and there's less, more variance explained, right? Less unknown, better predictive power, better ability to explain. Now, the two pieces when I look at the immune system and the breadth of the Human Immunome Project, what we really are seeking to understand is the variation in disease complexity and in our bodies, and the variation in population heterogeneity. Those are the two largest problems that remain in drug development. We don't really understand a disease, and even if we understood the disease in one particular instance, when we shift around the globe, it changes and we fail. It kills diagnostics, it kills drug development. So to me, when we generate additional data, and I would say it's really nice that we call it big data, but I actually think we live in a deep data world. So most samples, it's really expensive for us to collect samples, but now we can actually measure a lot of them. So it's actually a problem in computer science called P greater than N. There's many more features than there are samples, which basically throws a lot of standard machine learning and AI tools out the door. They don't work because you need to somehow bring in prior information to models. So within the Human Immunome Project, we look at this as we need to generate a multi-omic map of the immune system across the variation that exists. The immune system's variation, you need to study it across in a global fashion. And when we do so, we will be able to deliver a data set that will better allow us to explain that variation and then improve on diagnostics, improve on drug development.
Okay, so I think this is where I need to tell you that I am the executive chair of the Human Immunome Project, so I'm not here as just a completely innocent bystander, so to speak. But I also think we should take a moment to describe what these two different projects are and what the similarities and differences are. And so I think immunome is a subset of exposome, or that's kind of how I think of it, but
I would say
Long
it like
lost
this.
brother.
yes.
Yeah, no. You know, after, let's say, the human genome has for 40 years dominated biomedical research. And it was a fantastic investment. The first genome was 3 billion, but in the first 13 years, according to a Battelle study, it produced almost $1 trillion on revenue and created jobs in an incredible way. Yeah? But after these 40 years of the human genome, we know now that 5% of diseases are genetic, and about 40% have a genetic component. But it is assumed that 70 to 90%, especially of the chronic diseases, those which are really costly, which are plaguing us, have an exposure component. Yeah, lifestyle, chemicals, yeah, all of these together.
Stress.
Stress, yeah, social exposures, yeah, everything is included. Yeah, so the question is, are we at the point where we can tackle this systematically? Yeah? And this includes inflammatory reaction, and I'm not aware of any disease which has not an inflammatory component. Yeah? So this is a very important manifestation axis. And we believe that with AI, we have the enabling technology to crunch the data which we can generate. Because everything is exponentially growing. Data, AI capabilities, computer, but also chemical analytical methods. We can find in a droplet of blood almost every molecule. Our sensitivity has increased 1 million-fold in the last 30 years. Yeah? There's essentially nothing which escapes us. Yeah? And we can really find the stuff, and we can find what has changed in human biology and make sense of this by AI. And this is what the Human Exposome Project is striving for, to set up a similar global initiative to understand the exposure side of disease.
So what will you measure?
Most of this is done by changing the way you do safety science. In safety science, you typically start with a molecule, and then you characterize it and say, oh, this molecule has, let's say, a possibility to produce cancer. And then you would be looking into, would people be exposed to relevant levels? Get I proof that this is actually leading to cancer? In exposomes, you do the other way around. You look for interesting groups where clusters of diseases are observed. This could be groups of workers, this could be certain regions, ethnicities, and you ask, what is different in their blood, in their urine, in their biosamples, and you try to form an exposure hypothesis. What could have led to this? And then you work yourself downwards, and you bring in the experiments to find, is there a hazard associated which is really linking to these patterns. Yeah. So it's reversing this. I call it an inverse outcome pathway, because we normally look from adverse to outcome. Yeah.
Right. Cool. Okay. So I have more questions about that, but let's establish the baseline of what the Human Immunome Project is, and then we can start to compare them.
So the Human Immunome Project basically comes at this from the human side rather than the exposure side, but really in full agreement with the genomics, right? So amazing project, the Human Genome Project, but ultimately left us with a huge amount of unexplained variants and very far from clinical applications, right? When we look at what remains, that's the epigenetics, right? And the synthesizer of all that epigenetic information ultimately is the immune system, because this is the system in charge of homeostasis. And so what we're setting up to do is profile deeply the human immune system across the globe over time, longitudinal data sets. It's a complex system. You got to study it over time with interventions, perturbations, to see what happens when I vaccinate people and how that relates to the functional outcomes and so forth. Or any other kinds of interventions, and do so in a way that actually allows us to understand the relationship between the molecular high-dimensional measurements and clinical outcomes. So that basically means collecting the clinical phenotypes as well, right? And this, we feel, is key, because once I do the clinical to the molecular, I've shortened the path to translation, right? And second, once I've actually studied this across the globe in a multi-omic fashion, I'm basically in what you call the inverse adverse hypothesis or outcome, right? because rather than say, oh, what we need to be measuring in humans is what exists in electronic medical records, because in 1950, we knew how to measure it, like the CBC, the complete blood count. Instead, we're going to say, well, look, we've generated this high-dimensional data set. We can now associate what features we need to measure and are capable of measuring today to actually give the best predictive power or the best understanding of an immune state that and how it relates to exposomics, right? So this is kind of how we view the human immune system and the Human Immunome Project. And I think the two together are synergistic completely.
Yeah, so just to add on what Shai is saying, so the idea is we just announced a scientific partner network, which includes researchers on 5 continents, 10 different research institutes. In the United States, we have Yale, Stanford, and the Allen Institute. We have the Technion in Israel, University of Bonn, Radboud, and Leiden Medical Center in Europe. We have WEHI in Australia, RIKEN in Japan, and a site in Nicaragua, as well as 2 sites in Africa. We have Senegal and Uganda. And so each of these sites were selected because of the competence of the researcher involved or because of the cohort of populations that can be measured. We'll take some questions at the end, so if you want to hold it, maybe we'll answer it. But so keep in mind any questions that you may have. And so I'm curious, you know, so we're bringing these people, just to finish the thought, we're bringing these people together to emerge a process for clinical discovery, basically. So how are we going to test? What are we going to test? How are we going to reduce the dimensionality of the data to make this affordable? How will we make it robust? We'll come up with these standards, and then we'll deploy this in even more centers around the world. So that's going to be our process. I'm curious, Thomas, what your process is going to be. How are you going to discover these clusters, and how do you plan to address them?
I mean, the idea is growing, is mushrooming in different parts of the world. What we are missing at the moment is an overview on who is doing what and how we can make these things interoperable. So how can we report in a way that we can combine our data sets, for example? How can we bring in, at an early time point, governance and data sovereignty as well as ethical aspects? And this is what we just started. So last May, we invited some 400 eminent representatives of stakeholder groups by invitation only to Washington. We called it the Exposome Moonshot Forum, and we did not just have the scientists, the technologists, yeah, but we brought in the civil society. We brought in the big funding bodies. We had 4 NIH institutes, 2 in the organizing committee, OECD, WHO, UNESCO, the International Council of Sciences, the International National Government Science Organization. So there's a lot of these people who need to be involved in order to move something forward as a combined international global effort.
So I'm curious, are you involved with the One Health movement?
Yes, actually the name of our company is originally One Health, and we're doing a version of all of this more on the genomics, treatment and outcomes on the veterinary side and in the translational medicine space. And we're working with actually 1,350 vet clinics. Wow. From the one in Alaska to the university. We work with the Broad, we work with Stanford, and we have some Stanford here in the house. And so we're essentially already deploying targeted therapies across and gathering the data, correlating to any sophisticated diagnostics we can get our hands on. So very multi-omics, and looking for outcomes, for outsized outcomes. We already helped a human drug. This is public, so I can talk about it, for ASI. They had a drug hypothesis. They actually couldn't find an animal model. Like they really just could not. It was for sarcoma, and dogs get that abundantly. So this is like talking practically. And so they came to us, and we generated dog data against the drug. We worked with the Broad doing the human part, and our data on dogs was predictive. They moved the drug to Mass General. They're running many trials. It's in phase 2. So this is a public, like very tangible, you know, like, and this is how we're going to crack through certain diseases rarer cancers that, guess what, globally are not so rare anymore. So things are changing, and so it's a very practical approach. At the regulatory body, at the FDA, we're spending a lot of time on the topic of animal testing because the Office of the Commissioner, it's really bipartisan, by the way. This is not like, the administration's pushing it very aggressively. They want it as a win, and we are there, and we're actually part of the team that's helping articulate it. So we're very in the heart of the One Health in praxis.
Yeah, and so would you say that multi-omic mapping or modeling has accelerated our ability to step away from animal testing?
I mean, I'm a little bit of a different one, right? There's a lot of folks trying to bring in organoids, just different modeling. Because I'm bringing in a mammal that's co-evolved with a human that shares in the 86% the DNA with humans, that has, cardiac toxicity and liver. I mean, I'm bringing in like a real patient just for 4 legs. And we're healing that, like we're bringing in to really figure things out. So in that, we are bringing these human tools that you guys are and translating it to dogs, learning, and then seeing where does it converge with humans. Does that make sense?
Yeah, yeah.
And our data set, we use the Flatiron data set, initially Flatiron Foundation Medicine, for those of you who are familiar, as the initial starting point, layered our canine similar data set to learn and basically drive towards correlations of which drugs that we're not really understanding why are working so well and which ones actually not.
Is it hard to recruit canines?
It's totally weird. The vet space has zero. So I'm from Brazil. I was on the board of Planned Parenthood for the Western Hemisphere. I'm really interested in bringing care to that last mile that nobody wants to go. So I'm, with the voiceless, the dogs. It's really hard. So we're kind of recognized for being able to bring precision medicine to the absolute last mile, right? Again, whether it's Alaska, Singapore, Turkey, Brazil, go work with a vet, enroll the patient, not just help enable treatment, human-grade treatment, do all the sophisticated genomics, but then collect the outcomes. That's really hard. So with COVID, we saw the government got better at figuring out where the patients are, but being able to do that whole flow-through is really, really, really difficult. And that's the value of the data set and the uniqueness.
Yeah. Were you going to say something, Thomas?
Yeah, I mean, we have a tremendous problem of ignorance, what we don't know. If I take the example of toxicity, safety of chemicals, there's 350,000 chemicals which have been registered for commerce in the 19 most developed countries. 350,000. We have data for perhaps 10% of these, about 35,000, any data. If you go for cancer, to stay in this thing, 1% of these chemicals has been tested for cancer. And we will not change this because it does cost us $1 million for one chemical. It takes us 5 years, and we need 10 kilograms of this substance to feed 400 rats for 2 years. So this is nothing we can solve with an animal-based approach. For probably the most pressing question a consumer has, is this chemical you add to my food, to my cosmetics or my household cleaners, is this going to create cancer? Yeah? And this is why we need other types of tools, which have throughput, which have instant human relevance. I applaud certainly that a model which is based on natural disease in an animal is much, much closer than any artificially induced type of disease.
Yeah.
As a pharmacologist, I appreciate this. This is becoming
And
more
remember,
relevant.
you're studying a lot of the, the toxins and really our environment, which is really the truth, right? Like that's so important. And the dogs are, we can do studies, and for example, we know in certain breeds, certain dogs having an uptick of bladder cancer, there's a high concentration near golf courses. Near oil wells, like all sorts of chemicals, and the dogs are low to the ground. So it's like, it's a center. Basically, you can really interrogate questions that are just extraordinarily hard, as you're saying, either in a lab which is completely sterile and devoid of the world, or with humans takes a really long time and is a lot of money.
And maybe if I can add to this, I think the commonality here is, kind of, I said initially it's unknown unknowns, but really the way we're getting around it is, on one hand, measuring at high dimension, on the other hand, getting AI in, right? This has become an essential tool here. So if I think about the Human Immunome Project, I'm not just thinking about I need to generate data. I'm also thinking, well, there's going to be a limit to that data. There's going to be a lot of missing data. There's going to be a lot of things I don't sample. I need to ultimately really integrate this, even just for something as simple as handling batches, like technical batches. AI is going to help me. And so we're thinking about this together as going hand in hand. And I think this is true for exposomics. It's true for looking at cross-species differences. We went into this problem by saying, in my lab, we said, okay, people are studying animals for good reasons, whether it's for curing them or because they have no alternative to do so. How can AI come in and help out, right? We often humanize mice. That's a very long, expensive experiment that ultimately does a little to help. Well, could we bring AI to study the cross-species differences and build models that when I see a result in a canine or in a dog or in a mouse or so forth, I know better to expect what's going to happen in the human, right? And it's totally possible. It's just not a direction that people brought AI in yet. And I think this goes hand in hand in each one of those different things.
But first, you have to enable the AI. If you just see scientific work would be the thing to train AI on. Yeah, there's 3.4 million articles published per year in biomedical research. 3.4 million. But hardly any of this is open access. We have more than 50 million scientific articles which are medically relevant. 50 million. There's hardly anything the AI can access. We have to train it on the internet, which is a lousy source in comparison. The 500 million pages of scientific articles are behind paywalls, despite the fact that they almost always were paid by taxpayer money. Yeah?
Yeah.
And even if you have access to these papers, typically we don't have access to the data, to the raw data. And even if we have access to the raw data, increasingly, we have not access to the metadata, which in a standardized way tell you how the experiment was designed to produce this data. So what we really need is to change is the way we share information, the way how we report on our information, and who owns this, who can actually use this for training our AI systems.
So just to bring some of this together, because I'm actually working on this right now at the FDA. So we're taking existing drugs. So we're like a big repurposer. And I'm actually hunting. I'm even here at Davos hunting. If anyone here from GSK, Novartis, I want to talk to you. We're actually hunting for existing data sets. We're hunting for existing data sets in published work. We found some. And even within the FDA, they have the data, but inter-center, they don't communicate. So it's like really, and you have a commissioner talking about reducing animal testing and using data that already exists, but then even in its own center, like you can't access it. So I'm fighting that like right now, relentlessly. So we should talk later how to go.
Well, I mean, and even if you do have the data set, is it standardized in any way, shape, or form, right? Is there any conclusion that you can draw from that data, knowing that you don't know how that data was actually collected? Tagged
so
or
we
labeled?
get into that. So I need the raw data. The regulator won't accept just the results, so we need the raw data, and then we go in, we do an examination of the raw data, and we do a gap analysis, an analysis of what's missing, to then say, do you absolutely have to go and induce disease in a healthy animal to get data from a beagle that's young, has no comorbidities, which really doesn't recapitulate the human cancer experience in an aging population. So we're actually doing that right now with Amy Abernathy.
Yeah, cool.
She's
Who's one
on
of
my
your
board.
board
She's on my
members.
board, and
Yes.
she's like with us at the regulator.
Former FDA commissioner in the United States. Yeah, that's cool. So it's so fun to ask you questions I know the answer to, but so Shai.
I'll try and surprise with
Yeah,
them.
please do. There are a lot of data sets that the Human Immunome Project could tap into, right? There's proteomic sets, the metabolomic sets. There's lots of things that are constituent parts of what Human Immunome Project is trying to collect. Why don't you just import some of those into the models?
Why don't we just go to the old data? Well, I think the problem is that it's really in what it is that you're trying to achieve, and whether the old data is actually going to be a long haul that ultimately will not get you to that place, right? It sounds very easy to say, let's just go to existing published data sets, of many of which you can't get access to the metadata and so forth. Or maybe let's go to somebody who put a data set, kind of has a biobank and has a data set and has a lot of regulatory constraints on them and ethics and, already incentives aligned and so forth, and say, well, can you just lend me that data, right? It doesn't work. Ultimately, you can, when we think about science, the measurement, as difficult as it is and as expensive it is actually just a part of the way if you actually want to make impact. You need to get it all the way to healthcare policy and so forth. And so when we looked at this and we studied and tried to say, well, can we bring all the data together? What turns out when you're looking at the immune system is all the data that's been collected, it's actually, just a tiny fraction of what we think is actually needed, not even worthwhile to actually make the investment. And also, it's extremely non-diverse. So I've been running with Mark Davis at Stanford, I've been running the longest running high-resolution study of the immune system. Since 2007, we've been tracking a group of individuals annually and measuring all cool multi-omic technologies you can do that year, basically, right? That data set tells me a lot about people who live in Palo Alto, who I see some people from Palo Alto. So I would say that people from Palo Alto may be very interesting, but from an immunological perspective, they're kind of boring, right? And what we need to actually benefit the people in Palo Alto and everywhere else in the world is actually to get the global diversity. This has been true for genomics, right? We still don't have 50% of genetic variation exists in Africa. We're still trying to hunt down and get that sequenced, right? And it's certainly true for the immune system, and I would argue exposomics as well, which is basically you need to get that variation. So I need to decide where we invest. Try to get that data that's missing the raw, that's missing the metadata, that has all sorts of problems with it and so forth, or actually build it from the ground up the way it needs to be done, right? Data, multi-omic, collected with the metadata longitudinally. You can't study complex systems without following them over time. Otherwise, you end up in gigantic data sets that you don't understand what the differences are, right? So all of that information actually is easier to go from the beginning and do it well.
So I have to tell this story. There was a conference in 2022 where we brought together an extraordinary group of people and said, we think we can map the immunome and understand things about immunity that we haven't been able to understand in the lab. Do you think the measurement technology is there? Do you think the AI technology is there? What do we need to do? So I brought some AI people. There were some immunologists. There were some pharma people, academics. The AlphaFold team were there. And I thought, I would be translating for the AI people in between the two groups. And basically, the AI people show up and they go, we have the capability. Our models can do this. We don't know anything about immunology. Just give us your data. And the immunologist said, well, we don't have the data, and we don't know anything about immunology either. So
that's a good start to me.
Yeah, exactly.
Well, we know a lot about mouse immunology in a single time point and measured one piece at a time, right? Yeah. But if we want to do this in human, we need to do this right.
So this is a great lead into the question about, how have these measurements changed over time? How have the tools changed over time? How do we go from science fiction to being able to imagine like all of the different, impacts on our health and to actually be able to model them?
I mean, you've seen the prices, alone. The first human genome, $3 billion, now it's $200. We have seen, I mentioned already, metabolomics in 30 years, 1 million-fold increase in sensitivity. Yeah? AI, over the last 1.5 years, the price per token went down 180 times. Yeah? So it is an enormous amplification of what we can do, data-wise, but also in data generation-wise. Yeah? I'm particularly interested in these human-relevant test systems, organoids. We work with brain organoids in our work, for example. The number of publications in this field are doubling every 4 years. Yeah? It's all exponential growth. Yeah?
And I think from our side, I'm so excited about what you two are doing because we actually started really wanting to do the cross-species dog-human actually from the immuno-oncology angle. That's how we started, and then we lifted under the hood and it was like, wait, there's no tools to measure the canine immune system. We know it's different than the human, but how, like what could we do? And we were brought in by biopharma actually, who's desperate to understand cardiotoxicity, all different things about, basically amazing drugs that have such huge potential right now. They're not drugs, in 10 years, but it's really hard to test for toxicity, especially the biologics, the large oncology drug, large molecule drugs.
Yeah, that's really important. 1/3 of the new drugs is biologicals. Testing in animals is useless, because they are antibodies or against human structures or human proteins. There's very, very rarely they show any effect in any animal species.
You can caninize, right? So you really cross species, but the thing's it's never understood in the canine. What you guys are doing, the foundational work. I get you too. Sorry, I'm just learning about you. The foundational work that is like the Human Genome Project you were saying, decades ago now. So you're doing that, and I think so much is going to be built upon this. It's really important. So my 2 cents.
Yeah. I'll maybe add to that. Do we have time?
Yeah.
So I'll say I'll say that when we look at this problem, it's moving between species or kind of what is the goal that we need to get to. I actually I'm extremely excited where we're at, because similar to what you said about the price cost, if you look at the human genome, this is not. It started in 1990 and ended in 2003. At least from a marketing perspective, the genome got sequenced. Finished sequencing later, but the first 9 years we're developing the technology to measure to sequence at scale. And if you went in 2007, and I suspect it's the same for exposomics, and you said, can I measure the exposome? Can I measure kind of a human's immune system in a comprehensive manner? The answer is, get out of here, right? Science fiction, right? In 2007, I did an amazing science fiction experiment. I measured 38 proteins in the blood of a human, right? Of a group of aged individuals. You can now do in the 10,000 plus, right? We measured at Stanford, I was a postdoc there at the time, we measured 15 different immune cell types simultaneously. You can now do, basically hundreds of cell types simultaneously, thousands. The real argument is how many cell types are there? It's no longer an issue, right? At least 2 to the power of 40 or 80 or something as ridiculous as this, right? The technology to measure is there. The scientific problem of developing, can we actually measure something? That's not a problem. The problem is, can we scale it? Can we standardize this? Right? Can we build all the policy and the AI that actually is going to be able to do this huge operational challenges so that we can make the discoveries that change healthcare? And I think that's going to apply to both of our foundational projects, the verticals that we're taking, if it's in aging or looking in global health, which are things that the Human Immunome Project is related to, and the dogs will benefit. Right?
Yeah. Okay, so I had 2 questions that I wanted to still get to, and this gentleman in the back. Yeah, okay, so which one am I going to do first? I think we need to talk about money. So, when we start talking about the Human Immunome Project, particularly with family offices and so forth, everybody goes, oh, I want to invest in that, because they want to own the data and they want to own the potential outcomes of the data. But I'm curious, how you see the role of money in this scientific, and technological discovery phase that we're still in. Is there interest? Is it philanthropic interest? Is it for-profit interest? How is it changing? I'll let you take the lead on that, Christina.
Yeah, so we raised a venture. So essentially, my dad had cancer. I came here to the Cancer Moonshot launch and realized, oh my God, Joe Biden's son is has a similar journey. And thought we could bring in pet dogs and crack a lot of the codes that are known. There was a big meeting in Washington, D.C., kind of One Health Spirit, where I gathered all the folks who are really mapping this cross-species. And then I thought, my God, what am I going to do with this? Like, what's the business model? I have no idea. But luckily, there was a guy, Sam Altman, that now is famous, was not as famous, at Y Combinator, looking for people who wanted to map things and measure them in the biological world in order to, already contemplating AI, like building for AI, so AI native day one. So he initially funded us, and that was transformative. And then Andreessen Horowitz is our biggest backer. And they, and they actually are very tied to the administration now. So this link into the FDA directly, they're really catalyzing that. And that's been kind of a way to really jump from science fiction, nice idea, to interventions, optimizing interventions today. So I'm going to be doing a round soon, so I'll tell you more. But I do have an investor here, Bjorn. Okay. There you go.
Great. So Thomas, there's not a lot of economic incentive to do the work that you're talking about.
I mean, first of all, there is a value proposition in these methods because they are fast and can be run early on in the development process when you have little material. 1 day earlier to the market is worth $1 million or more for a pharmaceutical company. Secondly, the FDA, the NIH, and the EPA, they are changing at this moment. The FDA has announced a roadmap to move out of animal testing. The NIH has said they want now to focus on human-relevant methods. And what the FDA says is the most relevant thing in the world for pharma companies, because we are only 4% of the world population, but we are 53% of the drug market and 67% of the drug market under patent. All companies in the world only look for what the FDA is saying. And this is why at this moment this goes in. And with the NIH as the biggest funder in the world, going into these new things, announcing real-world data initiatives, these type of things are very promising that science will follow.
Okay. Okay.
Money. So I'll say, obviously, the value proposition for economic incentives ultimately of the Human Immunome Project is huge, and I think it's no one can miss it. The question is, how do you do this, and what's the right thing to do? And it goes back to the open science point that Thomas raised, right? Should this be a venture that's basically totally private and benefits a few, or would we actually, as a society, benefit more if this was ultimately an open access data that could launch 1,000 ships, right? And we've been taking that approach, that this data set must be open. Not only must it be open, it needs to be built with the right incentives for the players who are building it. So if I need to profile people in LMICs and go to Africa or East Asia or some other places where are not necessarily, don't have necessarily the people who would benefit, drug companies and so forth to benefit from this data, I need to think about how I actually don't increase the gap, but rather narrow it, right? And so we've been going with that approach, and I think, and this is the interesting news is, when you donate to these types of causes, when you come close to this community, right, you will find in your creative, you will find ways to ultimately benefit in initiatives that you would create on your own. So the $1 trillion that the Human Genome Project created, right, was doable, even though it was open access, right? And I think actually that society would have benefited way less if it was just the Celera genome sequence and nothing was ever let go.
Yeah, so Human Immunome Project imagines creating tools and platforms. We imagine new biomarkers for disease. We imagine new druggable targets. We imagine new reference ranges. And if you want to hear all about the impact of the Human Immunome Project, we have a panel tomorrow morning at 10 a.m. That Shai and I will do, where we can just kind of dig into that more fully. Well, I just want to say thank you to Emmanuel, to Tom Philback, to Science House, to Frontiers. It's so important to have a place to talk about science on a multilateral level and to bring people in from all these different countries. And, as I said earlier, as an ambassador for this institution that's being created, I think this is the red hot center of science in Davos. And And I just also have to put in a plug, the open science to solve global problems, such a good slogan, and
Yeah.
we are
And-
all big believers.
And a word on, who you are, Jane. She founded Wired magazine, sold it handsomely. Then she founded Chocolate. If you've seen it in Whole Foods, sold it handsomely. So, and now it's like, you're like science lady. I love it. I mean,
Thank you.
talk about somebody real leader that we need for this century.
Well, and she didn't mention she's a World Economic Forum Young Global Leader, so. Yeah,
But
there's
that was in
too.
2010
You're still young.
at heart.
Exactly. Anyway, I think there may have been more questions. We're not going to disappear immediately, but thank you so much.